Okay, we will get going. I feel like I'm in slow motion. My body's just barely starting to thaw. I had so many thoughts as I was going out to start my car, as my kids are in bed still sleeping from a cold day, right? I'm like, what? I got the text from ISU yesterday and I was like, sweet, we're getting one too. And then I read it and I'm like, oh, okay. I put on a long sleeve shirt. Then as I'm walking from my car, all bundled up, I put a face mask on because I thought I'm going to use one of these things. This could come in handy today, right? Then my eyes felt like maybe they were starting to freeze. And I thought, is that why my tears are so salty? To keep them from freezing on days like this? And then I thought of the Christmas vacation where the girls' eyes freeze as they truck out to the middle of nowhere to get a Christmas tree, right? I'm just like, oh man, this could be me, right? Holy cow. All right, welcome. I hope you all stayed warm. Some of you maybe were wise and are just watching from the leisure of your house with a nice warm blanket, maybe a fire. Shoot. All right. Any questions or concerns before I get going with this stuff? The exam is posted. Hopefully everyone's on there dealing with stuff. It closes today at 5. All right. We are going to start with this next chunk of material. And, you know, a couple of semesters back, I took the 1st through the 6th chapters and had them all as one unit because it kind of feels like they all fit together. But I also feel like that's kind of a lot of information and it's better to maybe separate it the littlest bit. So as we are moving along and thinking about the diversity, we're going to look at prokaryotes in this chapter. We're going to shift over and think about eukaryotic organisms in chapter 5. And as we get into the last bit, chapter 6 for this unit of material, I like to think that we're talking viruses, but it's a little more than that. There's some other things besides virus, some acellular pathogens that we will touch on. Lots of virus, but got to talk a little mad cow, right? So that's the idea as we progress through this chunk of material, is kind of more introductions to all of these organisms and some features about them. All right. So when it comes to prokaryotes, if we, throw the umbrella of both the bacteria and the archaea, these organisms, they are everywhere, I pretty much imagine. We like to say they're ubiquitous, pretty much everywhere, right? And I oftentimes think, where you think they are, you're right. And where you think there's no chance they could be there, there's going to be somebody that says, this is okay. I could tolerate this, all right? All sorts of crazy extremes, all right? Chiwanella in the deep oceans, basically barely trying to survive, but surviving, all right? And they've got kind of like the pili. These are some nano cables. I like to think that these are like little bacterial fishermen of the sea. As they cast out these nano cables, it's like little fishing lines that they're trying to seek different molecules of oxygen that they might be able to snag and in turn use for respiration. That's pretty awesome. So all sorts of interesting features when we think about the ocean with crazy microbes, some, living in super high temperatures where the, the black smokers as they are erupting, superheated water, high pressures, and thriving on stuff like sulfur as it's spewing out of there. So others here, halobacterium. This is, this is one that kind of gets me in biology, in science. All sorts of names get manipulated and changed and altered as we potentially recognize different things. Halobacterium was named this when it was still thought of as just some extreme bacteria, but they're really archaea. And in turn, it makes me wonder, why don't we switch up its name, right? Should maybe make a little more sense. These organisms are halophiles, salt lovers, and potentially extreme salt lovers. Living, maybe not living great, but surviving in salt that is so concentrated, it's basically just crystals, right? 5 molar salt concentration, which is super concentrated salt. They can survive, right? Pink Himalayan sea salt is the result of some of these archaea that are basically encased, right? Pretty interesting, right? I had a student, this was years back, that had a handful of different aquaria that he was pretty proud of, had all sorts of different saltwater fish, and he had used some pink sea salt stuff and started getting some of these blooms that you can sort of see here, where it kind of makes the water change colors, pinkish purple, kind of dependent upon who is really in there, all right? And although we're looking at the Dead Sea, the Great Salt Lake is a pretty good example of this. And you can see some of the color changes, especially in the summer, where the salt is a little more concentrated after some evaporation. Lots of big blooms of these organisms. So one type of extremophile, a salt lover, and pretty cool. These next ones play vital roles when we think about nutrient cycling in the environment. There are numerous organisms that help to provide nutrients to other things, like plants, which in turn we can tap into. Rhizobium are some nitrogen fixers. They live in root nodules and all sorts of different plants that we call legumes. So these, some clover, but beans also fitting in those categories, and in turn, basically producing their own fertilizer, really, as they're able to take the atmospheric nitrogen and convert it into usable nitrogen that the plants can deal with. There are other bacteria that do the opposite, and they break down nitrogen that's being excreted from us or through our dead bodies, breaking down these substances and releasing nitrogen gas back to the atmosphere. So it cycles through. And microbes are important in both sides of it. So this just another image of a different player, but similar idea with that. So there are all sorts of opportunities when we think about different places where we might be seeing these microbes. And deep down, a lot of this class, I'd like to think we want to think about some of these organisms that want to call our bodies home, whether it be on them or in them. In this case, potentially in our own, circulation. This isn't us. This is from a tick, but it would be going into us if it decided to take a meal, right? And we could see similar manifestations. These, and with the ticks here, we can see some of their cells, nucleated cells, with the rickettsia within there. That's what we're looking at, another blow up. Here's the nucleus of that cell and the rickettsia. So the hemolymph is like the tick's blood. Their circulatory system is an open circulatory system instead of closed like ours. So we mix it together, the lymphatic and the red cells, it's all one system. All right. In this case, we're seeing others. Now we could shift over and think about maybe some of our cells, where some organisms we might classify as they just have to be in cells. I mean, viruses technically might fit that category, but that's not, we're not on that chapter. We're not calling those living in the first place, but they need to be in a cell to get their job done, right? Bacteria, some of these fit those categories as well. Sometimes they don't have to be in there, but they could get into a cell and survive. In the case of chlamydia, they have to be in there. We call these types of bacteria obligate intracellular parasites. They have to be in a cell in order to replicate, in order to survive. They've got to be there, all right? We'll go through some details with this later, but ultimately what we're looking at here is just some steps in the life cycle as they are infecting and progressing through their replication inside of cells to proliferate to more. So these little elementary bodies infect a cell. Here we can see this cell getting infected 12 hours after infection. They're being pulled in through phagocytosis. They link up to the cell and it pulls it in. As it gets in there, they're surrounded in plasma membrane, and we call these guys reticulate bodies. It forms this big inclusion inside of the cell, and they start replicating. Over the course of now pushing about 2 days, we start seeing the cells that are ready to go on and infect new cells. And we call these, as we've shifted into the purplish ones, elementary bodies. So these are infected to the next round of replication. About 3 days into it, this cell is packed full of these chlamydial cells, these elementary bodies that in turn cause the cell to rupture and release the elementary bodies to infect more cells, right? Over and over and over, right? So it basically, replicates using, as it says down here, the host's ATP uses up all of the energy, creating more of itself, and then explodes it and goes on and infects more. Only a very short period are these little elementary bodies out before they're basically quickly pulled in to new cells. At this point, again, we'll hit some of this stuff a little later, but at this point, as they're out and exposed, that's when we might think about our bodies having an opportunity to kind of see them before they're back into a cell and hidden. So you might think about a little bit of an immune response being stimulated as these cells rupture and then kind of hidden away, and it causes it to decline. So you get kind of some fluctuations with that, stimulating and then hiding, kind of crazy. So this image, this is a bacterium called Neisseria meningitidis. This is a bad one, all right? This is a type of bacteria that causes meningitis, bacterial meningitis. There are a handful of potential culprits, all right? But as far as the bad ones go, this is number one on the list, all right? And if a patient is presenting to the clinic with symptoms of meningitis, whether it be bacterial or viral, there are some other potentials, but typically those guys, we start off treatment as if it's this, because if you don't, and that's what it truly ends up being, it's probably too late for that patient, right? Very rapid progressing infection. So more details with that to come. So these bacteria, the Neisseria, are a type of gram-negative cocci, and there are a handful of species of Neisseria, some pretty good pathogens like our meningitidis or gonorrhea that causes gonorrhea, both bad news. There are others that we might think about as maybe normal flora in your body. Neisseria Sika, I believe lactima is also normal flora in your mouth. Common to potentially make a smear and see some of those things, all right? And in turn, not be alarmed that maybe there's some bad thing in there. There could still be, right? But you might expect to see some of that normally, all right? So, and with that, let me flip it back to this plate. These colonies growing on here, this plate is called a chocolate auger plate. There are all sorts of different types of media. Blood auger plates are types of plates that have lots of nutrients through red blood cells that are incorporated into those plates. After the auger, after the nutrients and auger that's going to solidify it have been heated and sterilized, when it comes down to a lower temperature, they add some red blood cells. Sheep red blood cells are pretty common, but sometimes it's beef. They add it to the plate as it's starting to cool, and then those red cells, well, they basically turn it red, but it's full of more nutrients, and some bacteria really thrive with that. And in turn, we can also see some that break them down, those red cells, and give us some clearing, and we can classify some of that a little bit better. There are other bacteria that would like that, but they can't really break that stuff down on their own. Chocolate auger is blood, but instead of adding it after that auger is starting to cool down, we add it before it ever gets autoclaved, and it gets cooked up, boiled, and heated up, and those red cells explode, and it goes from nice bright red to brownish, and that's the chocolate auger. It's just cooked blood, all right? Now the cells that want it but can't really break it apart, we just give it to them, and they're like, oh, sweet, delicious, right? Now we can grow, all right? And some, like our Neisseria, grow very well if we give them opportunities like that. So this image, this is an X-ray showing some pneumonia, and we're seeing this caving formation as we're poking into this, looking, and occlusion as we're looking at that, but issues with that. And numerous, again, organisms have potentials for that, causing pneumonia. We will touch base on that later on. This bacteria, I think, is pretty awesome, and I'm pretty sure there are handfuls of videos showing some of these interactions. Should have been on it. Every time I flip to this slide, I'm like, oh, should have looked that stuff up. These squid incorporate some of these organisms in symbiotic or mutualistic relationship, a win-win, that allows them kind of opportunities to communicate with one another or to serve as maybe warning mechanisms. They can cause these bacteria to basically do some sort of advertising for the squid, changing colors, pulsating, lighting up or shutting off, turning on to scare something away, or pulsating to maybe woo a lover, whatever it might be. All right, these squid are using some of these organisms for all sorts of communication mechanisms, which is pretty awesome, I think. All right, so these are bioluminescent, kind of glow in the dark, as we're seeing here, without needing any sort of stimuli like shining a black light or something and making it glow. This just glows. All right, there are handfuls of like science outreach things that we try to participate in the biology building and try to show all sorts of different things. And we had an opportunity last year to bring back some of the science trek stuff where I think maybe third graders or third through fifth graders, I don't remember, young kids get an opportunity to come in, and it's kind of like night at the museum, although with COVID stuff, they stopped. They're like, we're not doing a sleepover, right? but it went to like 10:30 or something. So they would go around to different departments, different labs in biology and see things. And so we had a handful of different microbes with kind of crazy stuff happening. And some of them were these glow in the dark ones, which were pretty impressive, I thought. So not pulsating like this, but kind of like that, right? Pretty cool. I think we talked a little bit about Legionella maybe before. Legionella pneumophilia is a bacterium that we would think of or classify as like kind of a new player, an emerging pathogen. There are others like tuberculosis that we've seen for forever and thought we had a pretty good handle on, and now it's coming back, and we would call that guy a re-emerging pathogen. So Legionella has, I'm sure, been around for forever, but for it hurting people or causing issues to be on our radar, really pretty new, all right? 1970s was when it really kind of showed itself at a big convention, Legionnaires Convention, that infected a bunch of people, caused some fatalities, caused all sorts of detective work to be employed to try to figure out what's causing it in the first place, right? What's killing all these old dudes was the question, all right? They had no clue, all right? They didn't know how to grow it. They had to develop the media to get it to grow in the first place. And then ultimately backtracking and recognizing that it's like cooling systems that are prime locations for some of these organisms to thrive. There are other opportunities where they're just living out in the environment and not taking any sorts of situations to be brought to us. But when you give it a situation like this, the opportunity for it to be spread through a building is considerably more, right? They have recognized and isolated these organisms like in the misters at supermarkets. It's more than just these sorts of situations. So there are definitely some potential issues. Every time I see this image, the first thing that pops into my mind is hamburger, right? That's what I think when I see this, but it's not hamburger. But I'm like, sure looks like it is raw hamburger, all ground up and ready to cook, right? Hopefully you're not like ready to eat, because that looks a little pink to me, right? So this is an electron micrograph where we've imaged it and colored it up to make it look this way. And it's a gram-negative bacterium, so it should be pink colored. So there you go. Looks like hamburger, sorta, right? Not when you're looking at it under the compound microscope. It's not going to look like that, right? Salmonella, there are a handful of different types of salmonella, some of them that we call serotypes, that basically have subtle different immune responses where we could detect some antibody respectively. So typhi is one of them that causes typhoid fever. You guys know the story of Typhoid Mary? Maybe? Yes? No? What? Typhoid Mary, I know there's got to be some videos about her. I will see if I can find one and post it. She was a cook. I want to say it was New York, way back in the day. All right. And she worked for in the house of some prominent families and was their cook, right? And made some people pretty sick, got fired, got a new job, did the same thing. And finally people are like, what the heck's going on with this lady, right? And ultimately she was a carrier of this. Didn't really recognize she had any ill effects, but I bet you if you really talk to her, she's like, yeah, I always have diarrhea, right? Maybe not, right? But carrier, so maybe she didn't recognize any ill effects. but she's cooking all these people's food and really probably pretty poor hygiene. Hand washing, forget about it, right? I don't need to wash my hands. They're clean enough, right? Is kind of what I think. I think Semmelweis should have done a little talking with her, right? So that ultimately tried to run her out of town, but I believe she just progressed and kept cooking, right? When you got a passion, right, you just got to run with it, right? I gotta make these sandwiches for these folks, right? I have to, right? I don't know, right? All right. I'll see if I can find something that does that a little more justice than me. You know, you go to eat and you just hope, right? You just hope they washed their hands, right? I just assume they did, right? And deep down, they probably didn't, right? You're like, dang it, don't say that, right? Typhoid, marry me. All right. So myxobacteria, these things, I think are awesome. They look like little, super little mushrooms, but they're not, right? But they sure sort of behave like it, right? So the fruiting body, these little caps, the mushrooms, have a sporangium, which we would use a similar description when we're talking fungi, and they've got spores inside of them. In this case, myxospores, but spores, right? And ultimately allowing them to move and proliferate and grow as more little colonies or fruiting bodies. And I'm not sure what this is in the background that they're growing on, but I'd just say use your imagination and I'll move on. It looks like the dog trucked out to the yard and then here these things are growing a little bit later, but I don't know, right? I don't know. All right, Helicobacter. This is also one that's kind of like Legionella that we might say is like an emerging pathogen, something that's probably plagued us or some of us for a super long time, but really recognizing it, getting it on the radar as the culprit, pretty new, all right? So Helicobacter, I think, is pretty crazy. It can cause chronic gastritis. It can potentially lend to ulcers, maybe some serious ulcers, bleeding ulcers that are super painful, and also progressing to the potential for stomach cancers, right? Lots of individuals have been plagued by these organisms. There are all sorts of companies that sell antacids, and some of it potentially because of these culprits causing problems. There's other issues to warrant that stuff, but lots of people, all right, dealt with it, tried to treat with antacids or other things, and deep down, really the cause was this organism, all right? There were a couple of scientists that were pretty sure that this was the issue, all right? When you're doing science, you're not supposed to do science on yourself, right? You really shouldn't, right? Not a wise idea to do that stuff, right? It makes me think of other things. I'm like, well, how the heck you think you get Incredible Hulk, right? Doing a little science, right? Testing it out, right? You got it, right? Maybe it could be good, right? So these guys were pretty sure that Helicobacter was causing ulcers in people. They were able to get some isolated from a patient that had ulcers. They grew it up in the lab, got a big flask of it, I believe, probably poured each other a glass of it, cheers, and drank it down, right? pretty gross, all right? I think, right? They inoculated themselves by consuming this Helicobacter, and in turn, they both wound up with ulcers, right? They were able to cultivate it, show it, and say, Helicobacter is causing this. They did Koch's postulates, right? Kind of crazy. Something that, you know, ultimately is treatable and curable. And I think, you know, if you're gonna, I guess if you're gonna do some science on yourself, maybe giving yourself ulcers is probably okay, right? As in the big scheme of things, it could have been a lot worse, I'm sure, right? But crazy, right? There were some others that, you know, mites, little arthropods that, you know, are all over the place, truly dust mites and some mites on our skin, right? All sorts of different things. Dogs get some mites, get some ear mites, and sometimes it's pretty bad and they're always scratching their ears and whatever, and it can lead to other infections, whatever, all right? So not the same guys, but similar idea. The question was, are the mites that dogs get in their ears, are they an issue in people, or are they something that can't truly survive? So they got a handful of mites, slapped them in their ears, and come to find out they don't survive. It's a self-limiting thing. They only persisted for a couple of days, I believe, and then that was it. That to me seems a little bit disturbing, all right? I don't want to shove stuff in my ear, especially bugs, right? That seems wrong, right? What if they decided to survive, right? Oh man, pack back, right? I feel it, right? All right, the spirochetes, here we can see a handful, our little corkscrew organisms. They move spiraling through their environment by these axial filaments. Flagella twisted around itself that gives it its shape and its potential to spin through its environment. All right, there are so many images, so much stuff. When we think about our bodies, there are so many organisms that call us home, right? We are a crazy ecosystem for all sorts of weird or helpful organisms throughout our body, all right? Human Microbiome Project is something that has been sampling and evaluating and trying to get more of an understanding about all of our inhabitants, to think about those that are good guys, the normal flora, and to also get a grip on some that we could think of as the pathogens, those transient microbes that hitch a ride occasionally, or maybe some that are normal flora in one area that wind up elsewhere causing trouble. All right. So understanding details about that and getting more of a kind of census as far as what's really out there, what's on us, we're sort of just kind of still tip of the iceberg in really recognizing what's up. And that's pretty impressive, and kind of also could maybe be a little disturbing to know we don't really know a whole lot about what's really all there, right? Kind of crazy. So Bacteroides, these bacteria play huge roles when we think about part of our normal flora. They take up a massive amount of residence in our GI tract. They help with our metabolic needs as they help to break down some of our foodstuffs and release nutrients. E. coli is another that's right up there, high on the list, all right? Another gram-negative bacteria that high proportions, and something where we would think of as mutualistic relationships. We're both benefiting through these partnerships, all right? And more as we move through and think about some of these things. So they're thinking microbiome. There are researchers looking at differences in kids and trying to understand. These are things that I always find really pretty fascinating. You know, when you hatch out into this world, how is it that you are getting all of your inhabitants? How are you getting colonized, right? And it's not really until I got some kids that I recognized a lot of it, they're dirty little buggers, right? They put anything and everything in their mouths, right? Trying to figure out what's up, right? I don't know what this is. Let me taste it, right? Whether it's good or bad matters not. How do you know? You got to taste it first, I assume, right? So I think a good chunk of getting colonized with stuff is just flat out living and investigating the world around you. You're getting some of that, right? But you're also getting colonized potentially, not potentially, you're getting colonized before you ever even hatch out into the world. We're recognizing that what we thought were sterile environments really aren't 100%. There are potentials for that. And then as you are starting to come into this world, it truly depends on how you hatched out. Were you a normal vaginal birth, or were you a cesarean? Were you ripped out of your mother? Truly dependent upon how you made it into the world dictates some of those organisms that you may or may have not been introduced in transit out of her body, all right? And that sets the stage for some of your initial inhabitants, or maybe not, dependent upon what's up. And they've recognized that there are truly some differences between how these children came into the world, right? That's pretty crazy, right? With that said, they're looking at other things like potentials for behaviors. And maybe, you know, they say you are what you eat. Maybe it's the microbes within our guts that are truly driving some of that. And they say, you know, what I want is a donut, right? Or maybe not, maybe something else, right? Stimulating some of these things, or potentially other types of behaviors. That's pretty impressive. And, you know, there's a massive amount of nervous tissue within the gut, and potentially lending to some of these communications that stimulates that. I think that's pretty crazy. So these next ones, Planctomycetes, two different images, same but different, right? They may be sessile, so stable, anchored, or tethered in place, or they could be swarmers, where they can move, can propel themselves, right? With this flagella, we're not seeing all of it, but a little flagellum that allows them to whip themselves through their environment. So more root-like with this holdfast that allows them to tether to something in the water, and then maybe nutrients have started to become depleted, and they in turn move as they transition from the stationary to the motile form. This image, we're looking at a glass beaker or a cylinder. There are all sorts of ways we could generate these things. Graduated cylinders make pretty good containers for this. We're looking really at some soil and microbes all over it, all throughout it. This column is something that we call a Winograd column. And basically, we've got an opportunity to kind of return to our childhood and play in the dirt and make mud pies, stuff like that. That's really kind of what this is. You get some soil, you can potentially add stuff to it, some nutrients, different minerals, give it some sort of carbon source, and then you add a bunch of water to it and kind of keep it sort of like it's the soil in a pond, right? Is kind of what this is like. But it's exposed, as we see here. You oftentimes put it in a window, like a north-facing window, something that's not getting bombarded by the sun, and you let it go for a little bit of time, all right? A couple of weeks or a month, you can start seeing some stuff potentially around the glass. And the image isn't the best. You can see a bunch of light shining on this, reflecting away. There are parts of the soil that are potentially bright purple, right? With these purple bacteria, a type of bacterium that's got some pigments that it will secrete out and basically making it look purple. The green bacteria have some photosynthetic pigments within them, like some of the blue-green algae that aren't blue-green algae, they're actually cyanobacteria, these guys, right? And some other players, right? There are all sorts of other bacteria and other organisms, depending upon where you got your soil from, right? But proliferates within there, all right? And basically give it time for them to start growing, right? Till you see visible amounts of them, like growing bacteria on a petri dish. As you guys sampled stuff and put it onto plates, you'll see what sorts of stuff truly could be growing, right? This takes a little more time, all right? After a month or so, you start seeing it, and it can progress over the course of, potentially years. And you could look at different types of microbes. We started with the semester with the oil spill. Maybe you could get some soils full of some hydrocarbons, like some oils, and set up this situation. And over the course of time, if there were a few microbes that had the capabilities of feeding on that, they would propagate, and you would have a bigger bloom of them that you could isolate and tap into, right? If there was the hint of any of them being there, right? You're just giving them a greater chance of showing up. So not the same, but a sort of similar idea, were you to take that Winograd column and slap it right in the window, right? So it got all of the good, afternoon sun, it's going to get a massive amount of algae that starts to grow in there. It's going to cause lots of issues with the rest of the microbes in this column because it's going to outcompete them. And we've got a handful of situations here, a blow up of this waterway over here. Microcystis aeruginosa is a type of bacteria that, like I said, could be this, right? Cyanobacteria that's found in fresh waters and in situations where maybe through our own efforts, we lend to their population surging. So agriculture can be problems where runoff fertilizers making their way into the waterways provide extra nutrients, and the cyanobacteria can thrive in some of those circumstances, and you get these big blooms. And they compete with others and potentially cause the demise of all sorts of things, killing off fish and other stuff. Eutrophication is when you get these big blooms and basically sucking oxygen right out of the water in their process of surviving, starving the fish, suffocating the fish, I should say. Crazy. All right, the actinomyces, these organisms are kind of some oddballs as we're looking at it. This is an electron micrograph. They divide by a type of, I mean, it's still binary fission. They're still replicating that way, but we call it a snapping division. And it's kind of, it looks like a kind of a trap as they are replicating. This, not the best because we really only have one of them, but you oftentimes see a bunch of kind of little V's with them sort of falling apart from one another, tail to tail, if you will, as they're just about replicated. Then they exist as little singlets, all right? So that's actually the cyanobacterium that's doing this, the actinos over here. But this diphtheria is a nasty bacterial infection that hopefully you've been vaccinated against to prevent it. This last one on here, Gardnerella vaginalis, causes vaginosis. Here's a handful of cells and the bacteria all around it, and potentially able to detect this via a pap smear, showing the bacteria all over these cells potentially. And I believe, my memory doesn't fail me, when they see that, they call them clue cells, and it's indicating this. All right, that's my point where we will stop. I know we're right close to quitting time. Farewell. Stay warm out there.